In this lesson, what I would like to do is to look a little more deeply at the nitty-gritty details regarding translation. You now have a pretty clear understanding that it takes the messenger RNA, which ultimately gets read like a ticker tape through the timer. The timer is made of the ribosomal RNA, or rRNA, which has two parts, the small unit at the bottom and the large subunit at the top. Of course, there is also the tRNA. The tRNAs, of course, are responsible for bringing in the individual amino acids. And the goal, of course, is to lay down protein in a very, very specific sequence of amino acids. So let's move forward and take a look at some of the nitty-gritty details of translation. This diagram shows it in a very simplistic way. So remember from before that the peptidyl site is the P site, that is on the left side, and the amino acyl site, or the A site, is on the right side. What we're going to do here is slowly go through step by step the production of a fictitious protein that might be a couple hundred amino acids long. So here is our messenger RNA. It is being read by the ribosome. Remember that the first codon is always AUG, which codes for methionine. That is the start codon, and that is shown in the codon table. So the first codon is read AUG, and it's at the P site. So initiation has begun. At this point, the transfer RNA that is carrying the first amino acid, which in this case happens to be methionine, comes in to the P site. So the transfer RNA has pulled in the amino acid. It has lined up the anticodon loop, and that transfer RNA has lined up with the codon in the mRNA, which is AUG. The anticodon would be UAC. And of course, we have hydrogen bonds here holding the whole thing together. Next comes our second amino acid. Our second amino acid is carried by another transfer RNA. So our second codon in the messenger RNA is in order CUA. So it makes sense that the next anticodon complementary to that mRNA would be GAU. When you look at your codon table that codes for the amino acids, if we look up CUA, that's going to bring in a very particular amino acid. So that transfer RNA and the amino acid come in to the A site, as shown. So we now have two amino acids, the first one in the P site and the second one in the A site. And of course, the peptide bond is formed between the two. That's your amide linkage right there. And the process continues. The transfer RNA carrying the third amino acid comes in. And of course, it must be complementary to the mRNA codon CUU. And lo and behold, the anticodon in that third tRNA is complementary. Now, we don't have room for this third transfer RNA until something happens in the ribosome assembly. Very simply enough, the first transfer RNA that carried our methionine is now good to leave the ribosome assembly. And you'll notice that the first amino acid, which was attached to that transfer RNA, is now connected to the second the second amino acid in the A site. There's our peptide bond. There's our third amino acid. And now you can imagine that there has to be a shift. the second amino acid and the first amino acid, which are both being carried now by the second transfer RNA, has to shift to the P site. And the third amino acid, which is being carried by the third transfer RNA, can now enter the A site. Next, we have the fourth amino acid, which is being carried by the fourth codon or anticodon. So there's our mRNA codon for the fourth amino acid. It's CGA. The complementary anticodon would be CGU. And voila. What you can see here now is that the transfer RNA that was previously in the A site is now shifted to the right in the P site. And that gives room for the fourth transfer RNA to get into the P site. Notice that we're creating peptide bonds as we go. So we've got 4 amino acids now connected to the transfer RNA that is in the P site. Here comes the fifth one, the fifth transfer RNA, which is complementary to the messenger RNA, ACU, has the anticodon UGA, and the process continues. The third tRNA gets kicked out of the P site, and the growing peptide chain, or polypeptide chain, I should say, gets transferred to the transfer RNA that sits in the A site. And here we go. Fast forwarding, of course, once we get to the 200th amino acid, we have laid it down, perhaps, at this point, and the final messenger RNA that codes for an amino acid, in this case, would be GUU. Magically enough, as soon as we hit a stop codon, and remember there are 3 possibilities. In this particular case, the mRNA stop codon is UAG. That particular codon does not code for a transfer RNA that carries an amino acid. So in a sense, we have successfully produced 200 amino acids in a row to form our fairly short protein. To be honest, to give you an idea, insulin is only 156 amino acids long. That's a very short protein, to be honest. Albumin, on the other hand, found in blood, is over 1 million amino acids long. So it really is kind of mind-boggling to imagine that one could have literally millions of these amino acids connected to each other, kind of dangling off, if you will, the transfer RNAs on the A site and the P site. But that indeed is what happens. Obviously, if we don't have a stop codon, then the protein would just continue to be made, which would be a massively brutal mutation. Thank goodness that doesn't happen often. When it does, it's really catastrophic. So that is protein synthesis or translation in a nutshell. And of course, termination has to occur when we encounter a stop codon and the entire protein falls off. The ribosome transfer RNA mRNA complex. The end product is a protein, and of course that does give rise to the primary structure of proteins, which we learned in the first unit. Technically, the primary structure is simply the order of amino acids in the protein. That's really important. Insulin, for example, has to be exactly produced in the proper order to be functional. And if it's not, that insulin does not work properly. You could imagine that once we make this primary structure, this protein, once it's released from this assembly, will start to fold on itself and form the hydrogen bonds, the secondary structure, which remember are the alpha helices and beta pleated sheets, and then tertiary structure within subunits, and even quaternary structure where different subunits connect together. to form an active protein. So that, my friends, is kind of the end of the central dogma of biology. Yes, that's translation in a nutshell. I hope it makes sense. And once again, this picture kind of just shows in large scale format what we've been talking about. We've got transcription occurring in the nucleus. We have excised our introns from our primary mRNA transcript. It gets edited. That mature RNA or mRNA leaves the nucleus, where it literally gets attacked by this ribosomal tRNA assembly. And from there, we make our protein, and our protein grows and grows until we hit the stop codon, and end of story. This is another way of looking at what we succeeded in doing. Firstly, we had to start with DNA in the nucleus. It gets transcribed into messenger RNA in the nucleus. There's our mRNA, and you can see along this short section of mRNA, we've got the promoter sequence, which is kind of the recognition sequence at the beginning, which we'll talk about a little bit later. We've got our start codon right here, AUG, and the rest of those codons that we see here finally get read by the transfer RNA ribosome assembly until we hit a stop codon, and the result, again, is our primary protein structure. As you can see, we've got our methionine laid down at the amino terminus or the N-terminus end of the protein, and we've got our final amino acid, in this case, glutamic acid, at the carboxy or carboxylic acid terminus. That's it. Hopefully it makes sense to you. It's not an easy concept, but I hope that this video kind of made it a little bit easier than it would be if you just had to read the textbook.